Welcome to week 2. Today I want to talk about basic biological concepts, focusing particularly on the SARS coronavirus 2, and then I will talk about related viruses. I'll start by talking about coronavirus structure and function, reviewing a bit about what we talked about last week, what happens when you get exposed, and then tests for coronavirus and its responses. In general, coronaviruses are single-stranded RNA viruses. Their capsid contains lipids that help protect them from desiccation. Coronavirus 2 is one of 7 coronaviruses now that have been known to be disease-causing, pathogenic for humans. Prior coronaviruses that have had this property include human coronavirus 229, human coronavirus 43, human coronavirus UI, HKUI, all of which have been shown to be responsible for causing common cold. SARS coronavirus itself first became recognized in 2002 and was responsible for a significant outbreak, which we'll talk about a bit later, in multiple countries. MERS coronavirus emerged in the 2012 to 2014 timeline and was responsible for multiple clusters of illnesses around the world, although with a heavy focus in the Middle East. And now that brings us to SARS coronavirus 2. SARS-CoV-2 and this is a good reference for data, has a diameter of about 100 nanometers and a mass of about 1 femtogram. Those blue spikes on the outside represent proteins that are responsible for attaching to cells that they're ready to infect. There is protein in the outer layer of the virus that helps protect the interior material. And then the wound up material in the interior of that ball is the single-stranded RNA. On the upper right, you see comparison of the nucleic acid in SARS coronavirus 2 to the previous coronaviruses. A 96% similarity to coronaviruses that have been isolated from bats, a 91% similarity to coronaviruses that have been isolated from the animal pangolin, 80% similarity to SARS-CoV-1, and lesser similarities to the other coronaviruses. As a result of this similarity, many have inferred, although it's still remaining to be demonstrated, that SARS coronavirus 2 really evolved in bats and or pangolins, and then were transmitted to an intermediate species perhaps, and ultimately became the virus that infected people. Fairly small length in terms of viral nucleic acids, 30,000 bases. So 30,000 nucleotides in the RNA. And that encodes for about 24 to 27 proteins. Low evolution rate, which means the rate at which the nucleotides may change, and relatively low error rate. And so as a result, although there is speculation in the literature about different strains and so forth, there's really no evidence of substantial mutation that affects function of SARS coronavirus 2. When the virus encounters a human cell, it can enter it very quickly, at least based on what we know with coronavirus 1, within 10 minutes once it makes contact. It then undertakes metabolism or hijacks the cell machinery to undertake metabolism, which is really what I should say correctly. And after about 10 hours of doing so, it produces a large number of copies, perhaps as many as 1,000. So a virus enters in, 10 hours later, we get 1,000 viruses out. This is responsible for the explosive growth of viruses in a human cell. Host cells can be a variety of cells in the nasal cavity in the lung. Pneumocytes are cells within the lung itself. Macrophage are a type of white blood cell that is responsible for giving immunity. And mucus cells within the upper respiratory tract. And just comparison of the volume of host cells to the volume of a virus, the host cell volume, 1,000 cubic microns, orders of magnitude greater than the virus itself. People have actually measured the concentration of viruses within various human tissues. When samples are taken, we can get 1 million to 1 billion RNA copies per swab. When throat samples are taken, somewhat less. There have been very, very low, much lower concentrations of RNA measured in stool. Again, this is not for SARS coronavirus 2. The stool measurements at this point are very fragmentary, but this is perhaps an upper limit. And then finally in sputum. And as noted below, and we'll keep on coming back to this, measurement of RNA does not indicate measurement of viable infectious virus. So when the infection occurs, we can get antibodies in the blood, perhaps after 10 to 20 days. And again, based on coronavirus 1, although this is yet to be established for CoV2, we can get the antibodies to be measurable in the blood for 2 to 3 years. So that's what gives people a thought, and again, it remains to be proven, that antibody measurement can be indicative of some potential resistance. The viruses are fairly stable in the environment. There have been recent measures, specifically as SARS-CoV-2. The numbers here are a mix of data from CoV-1 and CoV-2, although the Van Dormalen data is for SARS-CoV-2. In aerosols, a half-life greater than an hour. In surfaces, a half-life 1 to 7 hours, depending on the surface, and both the aerosol and the half-life will depend on temperature and relative humidity. And those dependencies are still being worked out. Now, what makes SARS-2 so much more contagious than SARS is the fact that once you're exposed, you can enter a fairly prolonged period that may be several days where the virus is incubating, and even 1 to 2 days when sufficient virus may be emitted from an infected individual before symptoms appear. There are also cases where people can become exposed and infected being asymptomatic, and the asymptomatic ratio is not well known because of the lack of systematic random testing, but there's speculation that it may be that a quarter to half of the people who were infected might in fact be asymptomatic, which means that disease can be transmitted both by pre-symptomatic individuals in this phase and asymptomatic individuals who never proceed towards symptoms. And then recovery from mild cases may take 2 weeks, severe cases, 6 weeks. The fatality ratio's very much up in the air, probably a function of the nature of the population, the age of the population, and the adequacy of the healthcare system. But across the various countries, they've ranged from about 1% to 10%. Now, when you get exposed, what happens? And the next few slides are taken from a presentation from Karen Levy at Emory. First of all, the virus gets into your upper respiratory tract either by breathing through the mouth or through the nose. Viral replication can start in the upper respiratory tract, and then the virus proceeds both with respiration and with mucal flow down to the lower respiratory tract, and really focuses on the alveoli, the very, very small parts of this scheme, and the somewhat larger bronchi. And that's where the predominant respiratory infection occurs. In later stages of damage, sometimes a high level of infection of the alveoli can result in an accelerated immune response. And the response is called the release of cytokines. Now, without getting into the detailed metabolism, essentially cytokines are released to help combat foreign bodies that could cause infection anywhere within the body. But with the release of a large number of cytokines, so-called cytokine storm, damage to human tissue can occur. So you can get damage to the alveoli themselves, accumulation of fluid, sepsis, low blood pressure, and this results in the most common cause of death, acute respiratory disease syndrome. Other organ damage can occur because of the cytokine storm. There's been suggestion of cardiac and kidney injury, and a large level of inflammation throughout the body. One of the early signs of COVID-19 appears to be loss of smell and taste. And the biology of this has not been thoroughly described. It could be damage to nerve tissue in the nose, or it could be damage to other organs in the nose as well. As I said, a significant amount of transmission can occur in the window before symptoms develop. Now, this can proceed during the symptomatic period. This is a study of some earlier cases in China where they looked at the days since the beginning of symptoms. So CT value, remember, one of the ways to measure RNA is by PCR, by the polymerase chain reaction. And CT value, the lower that is, the lower number of cycles you need to get detectable RNA in the PCR reaction, detectable DNA in the PCR reaction, the higher the starting concentration of RNA was. So low numbers here represent high concentration. Very early on, after symptoms start, that's when you see the highest level of nucleic acid. And then as symptoms proceed, you get a decrease. And so presumably, even pre-symptomatic, there could have been a greater concentration of RNA and viral load. The viral load in asymptomatic patients, according to the study, was similar to that of symptomatic patients. So it suggests that asymptomatic cases can transmit as well as symptomatic cases. And it's really this that makes SARS-CoV-2 so much more explosive. Now, one of the things that gets asked is, with SARS-CoV-1, there was evidence for excretion in the stools. The evidence is still very spotty for this. These are really only the 2 studies that have been reported that addressed stool samples in infected cases. In the first case, children tested positive for CoV-1, or for CoV-2, excuse me. But this was only based on RNA, not based on culture. The second study was on adults, and again, RNA was detected in the feces for up to 5 weeks following onset of infection. So this leads to the possibility, and at this point it's only a possibility, that there could be excretion of viable infectious coronavirus 2 in fecal material and then in turn wastewater. However, we don't know, and I emphasize, just because you can detect it by RNA doesn't mean that it's viable. Here's another report of detection. And in this case, they used multiple methods in a large number of patients, over 200 patients. Again, a study out of China. And they detected by electron microscopy, and more importantly, by tissue culture, the live viruses. Although the concentration was very low, and this needs to be confirmed by other studies. In addition, the high concentration of electron microscopy detected virus, if those in fact were viable, raises concern for countries with poor health care and poor water and sanitation, water sanitation and hygiene. WASH is water, sanitation, and hygiene. Now, what happens when the virus is expelled by a cough or by a sneeze or even by normal breathing or normal speaking. First of all, the size of the virus itself, very small, 0.1 micron. But when it's emitted, it may be surrounded by mucus, saliva, other materials from the respiratory tract, and could be much larger, even up to 100 micron. The size determines what the lifetime is in the air before it settles out, and when it's inhaled by another individual who may be susceptible, where it deposits in the respiratory system, and could it potentially cause a case in another susceptible individual. This slide is from Lindsay Marr at Virginia Tech. And the principal methods for transmission of pathogens in general that could be via the respiratory route or by direct contact, and then touching hand to mouth. Could be indirect contact. Infected person touches an inanimate surface, and I introduced the word fomite last time. And then a susceptible person contacts the fomite, transfers that virus, transfers from hand to mouth again, or hand to nose, and becomes exposed. Large droplet transmission, which is what a lot of epidemiologists focus on, are droplets that would fall out of the air fairly rapidly, maybe 5 to 10 micron in size, but when they're emitted by the infected person, deposit directly on the eye, nose, or mouth and get taken in that manner. Aerosols are smaller particles that can stay suspended in the air for a long period of time, perhaps hours. And so a second individual breathing in from a space where those aerosols have been emitted may then inhale and become exposed that way. And people have actually measured aerosol production by simple acts such as breathing or sneezing. And in the case of breathing, the peak aerosol emission is about 1 micron. So remember we said the virus that causes COVID-19 is about 100 nanometers, 0.1. If these aerosols were to contain COVID, they would be a very small virus in a large coating of other material. Ordinary speaking, same thing. Peak of about 1 micron aerosol, and then much smaller amounts of the 5 micron. So, is it purely droplet transmission for coronavirus ID-19, or is it also aerosol? And this is a question that we'll be coming back to, I'm sure, multiple times in this class. Coughing, again, 1 micron, slightly less as a peak, but a pretty broad distribution of aerosols being emitted. So breathing, talking, and coughing release droplets that range from submicron to millimeter in size. But what size droplets really carry the virus? That's the critical question. And some of that for SARS-CoV needs to be determined. Now, we may be able to make inferences from a lot of studies that have been done with related viruses. Influenza virus is another RNA virus that contains lipid of similar size to SARS-CoV-2. And people have measured in experiments from breathing, from coughing, from sneezing, PCR measurable concentrations of viruses. Again, repeating the point, when I do that, it doesn't indicate viability. I showed you pictures of plaque last week. This is another picture of a viral plaque. To get viability, we need to measure the number of those clear areas on the plates, or a related technique is we culture the virus in a suspension of animal cells. We do it at different dilutions, and we measure the amount of sample that's required to infect 50% of these cells that are in the system. So for influenza, there have been measurements of those two measurements and measurements of those two results in paired samples. RNA copies per sample on the x-axis, essentially plaque-forming units on the y-axis. Lindsay Marr and the original authors of the study, which are Yan, suggested a weak but significant correlation between RNA copies and infectious virus. It may be a statistically significant correlation, but look at the R value. The R value 0.34, the correlation coefficient. So R squared is about 0.09. What does that mean? It means that only about 10% of the relationship, 10% of the variability in viable virus is associated with the RNA copies present. So I would say there's a weak but significant correlation, although the strength of the correlation is very, very small, and we have to ask whether it's useful or not. But in any event, Milton reported a study in volunteers who had the flu, and the exhalation of particles, both coarse particles greater than 5 micron and 5 particles less than 5 micron, as a function of wearing a mask or not wearing a mask. And what's clear here, two things. First of all, if we look at this bar versus this bar, which are without a mask, many, many, many more influenza gene copies, and the y-axis here is log scale, were emitted in the fine particle fraction as opposed to the coarse particle fraction. The second major finding in this work, which is shown in the graph, is that for the coarse particle fraction, we got a significant removal by wearing an ordinary surgical mask. On the other hand, for the fine particles, very, very small removal. So the bottom line to this is that, again, if in fact SARS coronavirus 2 behaves like influenza, surgical masks will do a good job of preventing droplets from being exhaled into the ambient environment if they're in coarse particles. But the coarse particles are the lesser of the amount emitted compared to the fine particles, and the surgical masks will not have as major an effect on that fine particle fraction. There was another study done with influenza virus in both fine aerosols and coarse aerosols. Again, this is RNA copies on the y-axis, now looking at emission without a cough and with higher cough rates. Somewhat greater, particularly at high coughing rates in the fine particles. Not so much difference with no cough or greater than one cough. So the bottom line conclusion is the majority of flu virus in terms of RNA copies are found in fine rather than coarse droplets and aerosols, which will lead us to ask, how did those particularly fine aerosols move around in the indoor environment? The other thing to realize is that depending upon the relative humidity and the temperature, even if we start out with a coarse aerosol particle, which will effectively be emitted at 100% relative humidity because it's coming out of the upper respiratory tract, which is saturated with water. As it persists in the air, it will be exposed to a lesser relative humidity and then decrease in size. And so we have a dynamic problem of decreasing size that we have to factor in with our transport relationship. And that occurs because of evaporation. So a 10 micron particle, even if it's large, may evaporate down to a 4 micron particle in the matter of tens of minutes. Now, I want to say a few words about testing. And this is a slide recently put out by the American Society for Microbiology. And it talks about the two main types of tests that are being used now for SARS-CoV-2 and how they might be used. The major testing where people are going in for all the swabs is testing for viral RNA. Finding nucleic acid in symptomatic patients says that they're currently infected, whether symptomatic or asymptomatic, with SARS-CoV-2. The value is to inform the individual of their status, take appropriate management of the patient, and inform others that may have been exposed to prevent transmission. So there's a beneficiary to the individual, to healthcare, and to public health. Antibody testing, which are apparently going to be shortly on the market, measures whether or not an individual has developed antibodies which would be necessary to combat the coronavirus, which typically would result after an infection, whether it's symptomatic or asymptomatic. So it's an indicator of past exposure. The value of it, presumably, is to detect susceptible individuals who are antibody negative. Identify individuals who have the antibody, but we don't know whether the presence of the antibody is sufficient to result in immunity if those individuals who are antibody positive become exposed again. And then finally, to also assist with contact tracing and surveillance. Again, there's beneficiaries to the individual, to the healthcare system, and to public health. Now, one of the issues with testing that we need to worry about is whether or not the test is sufficiently specific and sensitive. So if we had an ideal test, and let's imagine we had 200 people that we tested, 100 who had the infection, 100 who did not have the infection. Ideally, we'd like all those who were infected to test positive on the antibody test, or if we were using the qPCR test on the swab test, and all the 100 who were not infected to be negative. It's unlikely ever to get an ideal test. A non-ideal test might be we have the same 200 people, 100 infected, 100 not infected, but of those who had the infection, 20% were negative, are negative. Of those who did not have the infection, 15% were positive. So this would be a false positive. This would be a false negative. And what you need to think about is what the consequence is having an imperfect test to the individual, to society, if we make a decision and we don't have a perfect test. Now, some people have posed the idea of retesting as a way to get around that, which would work if the reason for false positives and false negatives were purely analytical. But if it were biological, intrinsic to the individual subject being examined, retesting would not really improve the situation. So we really need to understand what the false negative and false positive rates are in order to make a proper decision on using test information. Does measurement of the antibody predict resistance? Again, this is from Karen Levy's talk that I've modified. And the jury is still out. For SARS coronavirus 1, the immunity has been estimated to be about 3 years. In other words, you maintain sufficient immunity in your system to be relatively resistant for 3 years following its initial development. As of now, as far as I know, there's only been one study with animals using monkeys as a model, where they took monkeys who had been infected with SARS-CoV-2, recovered, had antibodies, and reinfection was attempted and was unable to occur. And this study was only a period of a month or so after recovery. So it suggests some level of at least short-term protection against reinfection. On the other hand, and there are questions with this data, but there have been reports of individuals who test negative for SARS coronavirus 2 after having recovered, but then appear to become positive again by testing. We don't know whether this is due to a testing error or actually resurgence of symptoms, and stay tuned for more data on this point. This is clearly going to be important to know if the idea of giving people, as has been reported in the press, some certificate of immunity is one means to talk about reopening parts of the US. Now I'll stop here, and then in the next segment, we'll talk about the two prior coronavirus illnesses that caused some level of global disease, SARS and MERS.